So I'll just say that our next speaker is Dr. Diana Deca.
Yes, so I will talk about grid cells in particular, and then I will try to explain how I think this applies to many other modules. So I think, in my personal opinion, the discovery of grid cells tells us something not only about the brain, but about the nature of reality itself. Because, like many parts of neuroscience, it works. Yeah. Okay. Okay. So I think that this works at the level of the interface between our understanding of the world and the world itself. It's not just something about information about neural data that we're getting, but also about the world. So I have yet another annoying technical. Yes, I also think that grid maps and other related data can be modeled to estimate how the brain maps space in general. And this has actually been done also in simulations and also in hardware implementations, because it's very easy to build a robot that moves in one way or another and then test all sorts of hypotheses. And that's actually really useful for neuroscience, because if then the robots based on that model are bad at navigation and they sort of hit obstacles and so forth, then we can always go back and gather more data and see what is wrong with our model and why the robot is not imitating human navigation well. So yeah, as Randall mentioned, in the past 7 years, I have been interested in whole brain emulation. So I was a very awkward kid when it started, and I sort of grew up with this topic. So my first paper was about for whole brain emulation. So I was trying to think about what tools are already there that we could probably make use of in order to achieve one day whole brain emulation. And then this sort of continued. I was trying to look at experiments that I think were useful and so on and so forth. Of course, there's no single answer to that. So this is an ongoing effort. Yeah, so I think one of the main challenges in our field is to extract causal information from the brain in a way that it can be emulated in a computer or robot, and not just implemented partially, but in a way that would correspond entirely with how humans behave. And while I think that some major advancements have been made in neuroscience towards that goal, ranging from the discovery of the action potential to cortical columns, to grid cells and their specific structure, I think there's no single model at the moment on how to best acquire and model this sort of data. So basically, I think we're still lacking a model of data acquisition and implementation that would really help us get causal information in a reliable way, as it is done, for example, in some branches of physics. I think this is my personal opinion. And while, of course, the correlation between, say, light or sound stimuli and spiking activities in neurons is well established, it still remains a correlational thing, because the causal connection between higher level stimuli, such as complex environments, learning, memory, decision-making, and neuronal spiking, which is the only readout that we really have from the brain, is still not entirely clear to me. So I just wanted to show you briefly what grid cells are, and I think this is really the easiest way. So this is a video of a rat where for each spike of that neuron that is being recorded, there will be a dot in this square. And you will see that the animal is kind of moving around randomly, and this will happen for a while. So now this is being put on fast forward, and you see more and more dots that still look kind of random in the box. But if you leave the animal long enough in that box, you will see some sort of pattern emerging that resembles a grid. And the grid becomes clearer and clearer in time. You'll see that, I guess. Yeah, so grid cells are place-modulated neurons whose multiple firing locations describe a periodic triangular array covering an open space, as you can see in this. And it has they have a very specific structure in that the grid spacing increases from one module to the next by a factor of 142, which is actually the square root of 2. And this enables the inner ring of the grid hexagon to double from one module to the next. And the grid size and spacing, so just the distance between these blobs, are given either by changing the speed-dependent head direction signal or by changing the width or radius of recurrent connections. So this is something we will discuss in more detail. But basically, grid cells are receiving multimodal inputs from head direction cells, speed cells, a lot of visual information, and so forth. And somehow all of these inputs combine to create this sort of grid. And yeah, this is interesting. So Brecht et al. Propose that there is a sort of isomorphism between grid cells and the external world. And this is what I mentioned in the beginning that might have sounded a bit weird to you when I said that when we're studying something like that, we're actually not just studying our own brain, but also our perception of the world within our brain. I'm not entirely sure of this, but what they propose in their paper is that there is a ratio of 1 to 2,000 between the grids in our brains and the external world. So they take an image of population activity in dorsal layer 2 in a patch of calbindin positive pyramidal cells. And they show different levels of isomorphism between that patch in the brain and the actual grid map that is created by the activity of the grid cells. And they describe that in a lot of detail. There are many, if all of these are coincidences, there are quite many of them. And it is an interesting paper to read. Yeah. So this is Immanuel Kant. He's a German philosopher, and he believed that humans could have judgments about the world that are synthetic a priori, which means synthetic means acquired through the senses, and a priori means acquired before experience. So it means that there was something in our senses that was independent of experience. So what he was trying to say is that we have intuitions about the world before experiencing the world. And to us now, this is not very hard to envision because, you know, some things are genetically embedded in our brains. For example, the ability to acquire language that was discussed by Noam Chomsky and others. It may very well be that we are built in a certain way to perceive space in the most efficient manner possible. But when grid cells were discovered, this became kind of a hype. First of all, the idea that there is isomorphism between the human brain and the world, and that there is an inherent structure to the world that we are somehow built to see. I guess that's maybe the best way of putting it. And another correlation that I noticed is with what the ancient Greeks called mathematical objects. So in a very poetic way, they suggested that there is an inherent structure in the universe and that it is our job to understand that structure. And parts of that structure were these figures that you see here. These are all the possible mathematical structures that there can be. And they also believe that in nature, everything we see is actually just an imperfect copy of that. So to some extent, to Plato, these mathematical objects were more real than anything else in nature just because of their perfect structure, while every other thing was a copy. And to some extent, the symmetry noticed in the grid cell pattern might suggest that this is actually true. So I'm going to explore that idea in more detail. First of all, it is possible for patterns to appear in a chaotic medium. And there are many examples of that in nature. And there's actually a very good paper from Alan Turing about the emergence of patterns in random systems. Yeah, and there was another interesting observation made in 2012, again about patterns and the grid-like structure of the human brain. So this was actually achieved with an fMRI scanner that had a resolution 10 times larger than that of a normal scanner. So people perform diffusion tensor imaging sometimes with fMRI machines, and in this particular case, they noticed that the nerve fibers in here had a structure that is very similar to that of a grid. So these are all random observations, which will all make sense in the end, I promise. So now we discussed about the existence of grid cells and the patterns that we see there. And now the next step in this process that I was talking about, so we had data acquisition, and now we have the modeling part of the neural data. So with grid cells, there are two main models that emerged. One of them is called the attractor network, which works a lot like a Mexican hat, and it just means that there are a lot of excitatory inputs to that one grid cell that are very similar to that pattern. So by receiving a lot of similar excitatory inputs, it's just going to have an output that is somehow summing up most of those inputs. And that center is kind of surrounded by other inhibitory inputs, and that creates this sort of Mexican hat-shaped pattern of activity. So that was one possibility. The other one was oscillatory interference, by which, for example, if we had a grid cell and it would receive a lot of place inputs and a lot of speed inputs, that would just be a pattern of spiking. And a lot of border inputs in a certain sequence, if all of these oscillations would then align in a certain way, they could in principle create a grid cell pattern. And what I think is very interesting about these models is that they don't only apply to grid cells, but to single neuron functioning in general. So this is maybe something that others could comment on later. But yes, and actually in time, there was one paper that kind of ruled the first model out by performing whole cell recordings on a grid cell and noticing that this model with a lot of similar excitatory inputs is actually not the case. Just by hyperpolarizing the neuron and seeing that the inputs are actually not mostly similar. So for all the Rick and Morty fans there are, I think the most interesting part is there are hardware implementations of grid cells and other cell types. I'm just gonna briefly go into my notes for this one. So, sorry. Duplicate. It's okay.
Windows key.
Duplicate.
Yeah. Let's just switch temporarily to Hangouts. Yeah. Yeah. Yeah, we'll do that.
Up to
For
this is fine. It's just
okay. Share back on that one. Screen share into boom,
share.
boom, back, and now we get your notes right up.
Thanks so much.
Okay, cool.
All right. Yes, so about hardware implementations of certain models of neural activity. So there is a group in Munich, Germany called GridMap that actually works on implementing some of these models into actual hardware. So the current hardware model that they have. Sorry? Okay. Gotcha. It says you're.
It says we are screen sharing.
Oh.
The wrong screen. Did you see the wrong one?
Oh yeah, yeah.
Oh, it looks like.
Yeah, it looks like it's working.
20 seconds delay.
Okay.
I think it's just a delay. Yeah,
Good.
definitely working.
Is called RADSLAM, and it serves as a model of how place grid and border cell interactions implemented in a spiking neural hardware, or Spinnaker, maybe you would know about this, made a package for neural engineering, and it's all hosted into one single mobile robot. And this robot uses spatial envelope synthesis, which produces grid, place, and border cells by interfering velocity to neural oscillators, because we mentioned before that oscillations, a summation of different oscillations is probably what happens with grid cells. So then the place grid and border cells in these models perceive theta oscillations at around 8 to 12 hertz from these so-called theta cells in the hippocampus. And this oscillation frequency increases when the agent's velocity is matched to the preferred velocity of a given theta cell. So without getting too much into detail, in this particular okay. That's the last one. Yes. This model is called RADSLAM, and they okay. This hardware implementation is called RADSLAM, and they used it in a very simple schematic in which they only had to code for cheese and home. They tried to include more visual inputs, and that's the funny part. The robot became extremely slow, and it had a speed of about, never mind. It was extremely slow just because of having to integrate visual input. So there is also a computational limit when it comes to integrating neural data and findings into a robotic implementation. Also, there was a bad version of that robot which used bats and their specific sonar system mapping, which also produces grid cells. I also think that there should be very similar activity patterns when it comes to memory formation, storage, and retrieval in the hippocampus. And there is a lot of evidence pointing to that, I think. One of them was a recent finding from David Tank, in which they showed that hippocampal and entorhinal cells also code for, in a learning paradigm, representations. So there are definitely other modalities that are represented there, because important is to find a way to represent that data in a way that is easy to visualize. I think that's why I wanted to use the case of grid maps for that. So yeah, just to sum it all up, in case this has been too much information, I think that there's a very simple model for whole brain emulation, which involves acquiring neural data in a certain way. And in this case, it's just getting grid maps from electrophysiological recordings and modeling them. So in the video, then the next step is, of course, creating computational models for that data, which in this case has been attractor networks or oscillatory interference. And finally, the third step is a hardware implementation of the modules that can be implemented into hardware. And in the case of grid cells, was using only oscillatory interference because it was observed in neural data that the first model of attractor networks is actually not viable. And so the main observation from the hardware implementations of grid cells was that there has to be less inputs to allow for sufficient memory recall, because that allows for mapping a space efficiently. And less inputs in this case just meant less visual inputs. I believe that this is a feedback loop, and I don't think that whole brain emulation will appear instantaneously after acquiring enough models or enough data. Rather, I think it's an ongoing process of constantly improving the way in which we gather data and make computational models out of data into hardware. And I think here there were some good examples of how that can actually be done in the future. Yeah. How am I on time?
Probably fine on time, because as it turns out, this session has way too much time in it, and Mikhail is really only going to be ready.
We can keep on discussing then. So yeah, to conclude, I think these neuroscientific discoveries they don't just tell us something about the neural correlates of a subjective perception or navigation or anything else. Context independent, given their context independent nature of grid cells in particular, they actually impact on our understanding of nature, of the nature of reality in general. What I mean by this is that their discovery might have informed us about certain innate categories that we might be having, although this made people test that hypothesis even more. And what they found is that when certain objects around an arena, the grid map actually over time, which means that these representations are in fact not entirely a priori as we initially imagined. Rather, I would imagine these cells as, or at least the grid that is being put onto a certain space, as more like a blanket that is thrown over a space rather than unmovable. Right. So I think that pretty much sums it up. I'm trying to think of other examples where this happened. It would be nice if there are suggestions. I think in the end, when it comes to neural data, this is just something I drew when I was bored. It is not easy to categorize things in a certain way, and it's something that people take for granted very often. When categorizing something, it then becomes very easy to put it in a certain box. And we have, for example, the concept of noise, and what happens most of the time is that noise is just something that we didn't fully understand or categorize yet. So my hope is that in the future we will find ways to represent memory formation or decision making in a similar way as we represent grid cells. That is very obvious of direction selective cells or place cells and so forth. So with that, I again apologize for the technical difficulties, and I'm very happy to take your questions.
Is it okay to just stay on your side?
Sure.
Okay. Excellent. Definitely, there are some questions, and since you dove into both philosophy and neuroscience and robots, there are questions of all kinds. The first question I'm just going to pull up here, which is not the one that Mallory just posted, but a slightly different one, and asks, how do grid cells work on blind specimens? Has that been tested?
So, very interesting question. Not from what I have heard, but it has they have been tested on different modalities. So I think the case of bats that are actually blind and use a sonar system to map spaces shows that different modalities can be integrated in a way that still creates the same pattern. The question.
Thank you. Now I'm going to mention the one that that Mallory put in the chat box here. It's actually also one from the forum. And the question is, you mentioned correctly that Kant, a intuitive, in this framework, and given that the world would be a kind of projection of these a priori patterns, consequence to whole brain emulation research in the current philosophical discussion. Ooh, that's a big one.
Wow.
How do you interpret that?
Question, because I think it's similar to how I frame my talk. It includes a lot of elements. So let's try to parse it. So whole brain emulation, how can we achieve whole brain emulation considering the concept of synthetic a priori judgment? Well, in principle, I think the level of the emulation, but I think that we should be able to aim results if the emulation is done well. That would actually would just show that the emulation has emergent properties that we couldn't fully understand before creating it. Yeah, which is actually another important point. We can emulate things that we don't understand. That's theoretically, yeah.
Yeah, actually, my comment adding to that is actually, I think if there are a lot of these a priori patterns or things that are common among, let's say, all of us, all animals, all brains, always in the same brain region, etc., because there are these a priori patterns, that should simplify the task, right? Because if you can get data that tells you something about these patterns or about these forms as they relate to a particular brain region and how it operates, then you can use that as for how you will build the models for a specific brain or specific person. So it sort of simplifies the problem as opposed to a completely open search space, right?
In a way. So that's a very interesting point. I just wanted to add that, so Immanuel Kant gave mathematics as an example of a synthetic a priori. Yeah, it's kind of hard to connect the concept of how mathematics emerges in the human mind with neural spiking. I think that's an issue that when discussing whole brain emulation, how do we, I definitely agree that it should be easier to do that, but yeah, I think the two can be connected.
Let me just quickly switch to the audience right here. All too tired? Yeah.
Anyone
The
awake?
survivors are still there, but they can't formulate questions anymore. Then let's see. Yeah, I don't know. Okay, one of these things wasn't really a question. It was more trying to understand your graph when you had the sort of this cycle, right? Back to from hardware back again to grid cells.
Right.
I was trying to understand how what's happening there when you're going from hardware back again to grid cells.
Yeah. Sorry for not being able to open the slide again. Arrow from the hardware implementation was going back to data. In the case of grid cells, this was a good example with the fact that we computational power to be able to integrate enough visual information for a robot to move around a room fast enough, and also to create the same patterns, because that means it has to slowly form a memory of a place. To data, I guess they try to simplify the entire experiment to better test data in the future. So it's definitely a feedback loop, I think.
I'm imagining Roombas driven by grid cells at the moment.
Really cool, yeah.
Yeah.
Question about.
You want to speak into this?
Sure.
So question about that slide where you had in a brain and then building a computational model and then implementing that in hardware. And how would brain work or paradigm do you think that is for research? And do you think the actual hardware is versus just kind of going back and forth between models and data?
So I think that's also a very good question, because there are some modules where I might have trouble with implementation, for example, olfaction or some other ones. I mean, when it comes to navigation, this is very easy. Of similar modalities, memory is easy to imagine because that's easy to test in behavioral experiment. Olfaction, I'm having trouble imagining that many modalities which wouldn't make use of that. Depends on the state of knowledge. Of course, for single neuron models, there's not necessarily a need for hardware implementation unless the ion channels opening and closing in response to different situations. Yeah. Quite fun.
There's another question coming in from the online platform. This is by Chris G. In the relation between theta pathway and grid cells, please.
I'm not sure. So this is probably a good question that I'm not sure I fully understand what a theta pathway is. In my talk, in the context of oscillations and these theta rhythms summing up to create a certain pattern. Yeah. Could you repeat that question? Is that theta pathway and?
Theta pathway and grid cells.
Yeah,
What is meant by theta pathway? Perhaps the way theta is generated or the way that theta is used throughout different areas.
If it's a theta rhythm, it definitely contributes to the grid maps that entorhinal cells have. Not entirely sure I can. Yeah.
Go ahead.
Can you? Oops, sorry.
My question is, place cells represent various topological maps across space. So when I think about the entire Bay Area where we are, how do my place cells, are we talking about place cell accommodation across to accommodate any scale? Like I think about the planetary system and I have place cells for that, or is it literally just the visual system and me doing motion planning within my current environment?
Can you really ask about place cells, not base cells?
Either or, yeah. I mean, we should.
Fascinating question in itself, and I guess it's also about the extent of mapping, right? Which I would imagine is memory dependent. No, there are many questions about. I think studying the brains of birds would be very useful in this case because they manage to find their way every time. So they probably have a large-scale representation of the space they map when they fly over, and then a smaller scale. I'm really speculating now, but I think these maps seem to become clearer and clearer with experience, or as the memory of them becomes. So I think other maps become. Some maps are faded, and specific maps of specific places, like the bird's nest location, becomes clear in time, something like this. It's same with the Bay Area. That's how I would imagine that if you drive, you have a certain representation of the Bay Area that is very much related to your car rather than walking.
I don't know if we have any more questions on this topic. The forum seems to have run out of specific questions for this right now. Well, in the audience, I see that Mikhail Lebedev is already ready, so I could go on and introduce him. Are we there? I think so. Thank you so very much. This was awesome. And we are going on to our last speaker of today, Mikhail Lebedev. Mikhail is a senior research scientist at Duke University, experience in neural interfaces or brain-machine interfaces, responsible for a number of special issues and topics closely related to the topics we discussed today. Free to begin sharing your screen, and if you'd like to say more about yourself, feel free to do that as well. And we look forward to.
Okay, thank you very much. Can you hear me?
Yes.
Do you see my screen?
Yes, we can hear you well and see your
What
screen.
about my screen?
Yeah, Ara can do that here.
Yeah, indeed, we can see your screen.
Just turn this up.
Perfect. Okay, so I will talk about representation of multiple behavioral variables by cortical neurons. Yeah. And let me start with showing you this picture of typical brain-machine interface, but it also applies to any kind of reading from the brain or decoding information, or maybe like reading from the brain and writing somewhere. So here is the brain. Then electrodes are implanted in the brain, and information is recorded, decoded, and then sent to this prosthetic hand that performs writing. So this is a typical design of a brain-machine interface nowadays. But I will argue that brain-machine interfaces or any kind of decoding approaches can work better. So what do I mean by that? I mean that we can try to decode multiple things from the brain. So there is a story about Julius Caesar, that he was able to do many things at one time: write, dictate, read. So modern people also can do this. So why don't we try to decode this information from the brain? So for this, monkey brain is most useful because, first of all, monkey brain resembles the human brain. It has basically the same organization of as the human brain, and it is also easily accessible with invasive recordings. So we can try to implant electrodes in different parts of the monkey brain, and there are many interesting parts in the monkey brain, like here. There is a motor cortex with leg, arm, face representation. There is a somatosensory cortex, a prefrontal cortex where the monkey's thoughts are represented, premotor cortex, eye fields, etc., etc. So you can put electrodes in different parts of the brain and then try to decode information. So this invasive approach was pioneered in the 70s by Edward Evers. And what he did, he would have an awake behaving monkey, and then he would insert an electrode in the monkey brain. The monkey wouldn't feel any pain or anything because there are no pain receptors in the brain. And then the monkey would do a motor task, and Evers would record typically one neuron at a time, and he would get neuronal responses like this. But here, this is a later work of Georgopoulos. Here, the monkey moved a joystick in different directions, and you can see that this particular cell was very active when the monkey moved the joystick to the right, and then the cell was not very active. Actually, when the monkey moved the joystick to the left, and the cell was not active when the monkey moved the joystick to the right. So an experienced neurophysiologist can just listen to the cell firing through the speaker, and he would be able to decode information just by listening. So it is important for this kind of decoding that the monkey may not even do any overt behavior, like in this work by Krement and Kalaska. So the monkey was shown a target, and then the monkey was preparing to move but was not allowed to move. But still, by looking at premotor cortical activity, they could tell that the monkey was really preparing a movement in a certain direction. So what kind of variables, multiple variables, can we decode from the monkey cortical activity? So I would list the following: motor parameters, orientation of attention, gaze angle, memory, time. So let's consider this example from sports. This basketball player looks, attends, and performs a movement using the same spatial location. Here, gaze, orientation of space, spatial attention, movement goal, they all colocalize. And this often happens, that there's quite different behavioral variables, they colocalize to the same spatial locations. But they can also be different in space, like in this example from hockey, where the hockey player looks at the puck, prepares to pass the puck to his partner, and at the same time he attends to the player from the other team. So here, gaze, movement goal, attention, they all have different spatial locations. So, like, can we mimic something like this using a monkey experiment? Yes, we can, and I've done this. So, yeah, the experiment looked like this. The monkey was sitting, and then in front of the monkey, we placed two feeders with very tasty food pellets. So one feeder was called stationary feeder. It was stationary, and I could move it just manually to different locations around the monkey. And actually, the monkey could reach it, but this was not allowed, because if the monkey attempted to reach for the pellet, then the pellet simply disappeared. So we also had a robot feeder. This feeder was mounted on a robot that could move around the monkey. And by the way, this robot's name was Guard, the name taken from the movie, The Day the Earth Stood Still. And Guard actually moved back and forth relative to the monkey. And when Guard entered the zone that the monkey could reach, like this, then the monkey was allowed to reach. And the monkey had two tasks. Task one is when the monkey reached to the feeder mounted on the robot. And in task two, when the robot entered the zone that the monkey could reach, the monkey didn't reach to the robot, but instead it reached to the stationary feeder. So once again, two tasks. In both cases, the monkey has to look at the robot and attend to the robot, because when the robot entered the reachable zone, in one case, in task one, the monkey reached to the robot, and in task two, the monkey reached to the manual feeder. So in task one, the monkey attended to the robot, and also the robot was the goal of movement. In task two, the monkey attended to the robot, but the goal of movement was different. It was the manual feeder. Additionally, we looked at eye position, and the monkey had a tendency to alternate eye position from looking at the robot to looking at the manual feeder. We placed the robot and the manual feeder in 4 locations around the monkey, and these are called here far left, near left, near right, and far right. So 4 possible locations. And this configuration of the manual feeder and the robot feeder can be simply described by a 4x4 matrix, where columns of the matrix represent the position of the robot feeder, and rows of the matrix represent the position of the manual feeder. There is nothing on the diagonal because physically we couldn't put the manual feeder and the robot in the same place. So now let me show you some examples of our recordings. And once again, this 4x4 matrix. And here is a neuron that was sensitive to the robot position. Why? Because when the robot was in the far right position, this neuron was very active. And when the robot was in the far left position, this neuron was not active. Now, if you look at the changes in this neuron's activity with the position of the stationary feeder, they were very small. So this is a neuron that represents a robot position. And we can also use color coding to show the same effect. Here, red color is high activity of the neuron, and blue color is low activity of the neuron. So, and here you see a vertical band, vertical red band, which means that this neuron represents the robot position. Now, and also we recorded some neurons that had horizontal bands, meaning that this neuron is sensitive to the stationary feeder position. Now we can look at different neurons in different situations. Task 1, task 2, then depending where the monkey looks, and also when in time we analyze this neuron's activity. So, like, trust me that this particular neuron represents attention. Basically, why? Because it has this vertical band practically in all cases, except when the monkey in task 2 is reaching to the manual feeder. So when the monkey's attention disengaged from the robot and is now at the manual feeder. So, and also, even if the monkey looks at different locations, still this neuron represents the position of the robot. So this is an attention-tuned neuron. This neuron represents gaze angle because when wait a second. This particular one represents target tuning. So, because in task 1, this neuron has these vertical bands. So when the robot is the target of movement, the bands are vertical and represent tuning to the robot. But when the goal of movement is the manual feeder, then you can see these horizontal bands, meaning that this neuron is now tuned to the manual feeder position, which is the goal of movement in this task. So this neuron represents the target of movement. And finally, we observed many neurons that represented gaze angle. So basically, if the monkey is looking at the manual feeder, then this band is horizontal. If the monkey is looking at the robot feeder, then this band is vertical. So we recorded in different places in the frontal cortex, and we found the places were dorsolateral prefrontal cortex, frontal eye field, dorsal premotor cortex, ventral premotor cortex, and supplementary motor area. And the main finding was that the majority of neurons represented the attention, not the motor target. Even in areas like premotor cortex or supplementary motor areas, which are considered to be motor areas. And also in places like prefrontal cortex and frontal eye fields, we observed many neurons that represented gaze angle. So basically, this shows that when we give a monkey a task that disengages different behavioral variables, we can identify the properties of neurons much better. And in this case, we found out that many neurons represented orientation of spatial attention, and if a simpler task was used, they would be characterized as representing some motor parameters. So let's now consider another situation. So in this situation, orientation of spatial attention was compared with working memory. So in dorsal prefrontal cortex is famous as the area that represents working memory. But working memory experiments typically do not disengage orientation of attention from what the monkey is supposed to remember. So we conducted this experiment where we disengaged the orientation of monkey spatial attention from the spatial location encoded by working memory. Here, the monkey started by looking at the center of the screen. Then, at a peripheral location, a round target appeared, and the monkey was supposed to remember the location where this target appeared first. Why? Because a little bit later, the target started to rotate and ended up at a different screen location. The monkey was required to attend to this target. Why? Because if the target dimmed, then the monkey was supposed to make a saccade to the target. But if the target brightened, then the monkey was supposed to make a saccade to the location where the target first appeared and which the monkey was supposed to remember. So here, once again, the results could be represented by a 4x4 matrix because we used 4 screen locations for the remembered location and for the attended location, and also they could coincide. So now there is data on the diagonal. So some examples. So this prefrontal cortex neuron represents attention tuning because we can see this vertical band of activity. So this neuron represents where the target is currently located and where the monkey attends, but not so much what the monkey remembers. Here is another example, and this neuron represents memory because you can see this horizontal band of activity, and so this neuron represents what the monkey remembers, but doesn't care about where the monkey attends. But also we observed neurons that represented both the attended location and remembered location, like this one. That has both a horizontal band and the vertical band. So neurons like this we called hybrid tuned neurons. Now, if you count the proportions of neurons of different types, then the majority are attention cells, then the minority are memory cells, and the 24% are hybrid tuned neurons. Since we have both attention location represented by prefrontal cortex activity and remembered location represented by prefrontal cortex activity, then we can try to decode these two variables. And here I used a very simple linear decoder to decode both locations, and you can see that the locations could be decoded. The attended location was decoded very well, whereas the remembered location was decoded not so well, but still with reasonable precision. Also, when the target travels on the screen, we can apply a linear decode and decode this movement of the target on the screen. So let's now consider a different experiment, but also that has two variables that will be decoded, and also this 4x4 matrix representation will be used. Here, the monkey had a joystick, and it performed a very simple task where it moved the screen cursor to different screen locations. So the target appeared on the screen, and the monkey moved the cursor to that target. But there was a caveat in this task. Here, the target appeared, but then in 50 to 200 milliseconds, it could jump to a different location. So the initial appearance of the target in some trials was just a distractor, because the true target appeared later. So here, once again, we represented the data using this 4x4 plot, and the distractor location is represented by the columns of this matrix, and the true target locations are represented by the rows of this matrix. And if you examine this particular neuron carefully, you can see that initially it responds to the distracting target, but then when the distracting target disappeared, then it switches to representing the true target. And so initially there is a vertical band, and then later there is a horizontal band. And color-coded diagrams can be used for the same purpose. And it is interesting how these bands develop in time. So when the distracting target is shown, you can see the appearance of this vertical band. Then this target disappears, and the true target appears, and then there is a conversion of this vertical band to a horizontal band. And finally, the most clear feature is this horizontal band, but the trace of this initial vertical band is still there, even though there is no initial target on the screen, and the monkey even moves to the true target. Now another example of an experiment where two things are extracted from the monkey brain at the same time. Here the monkey had two joysticks, and it controlled two virtual hands on the screen, and these hands could move independently to their designated positions. And also we conducted the same experiment using a brain-machine interface design. And in some experiments, the monkeys continued to operate two joysticks, and in some they had no joysticks, and they controlled the movements of these two virtual hands simultaneously and directly by brain activity. So once again, here the data could be represented by a 4x4 matrix, where columns are the target for the right arm, and rows are the target for the left arm. And we got very interesting results. They were quite complex, so I will not go into all details. So for both motor cortex, primary motor cortex, and for supplementary motor area. But let me just show you a video of how the monkey performed this task under brain control. So the monkey starts with positioning the virtual hands at the central targets, and then two peripheral targets appear, and the monkey moves the virtual hands to these peripheral targets. Since the peripheral targets can appear independently in different locations, this is a type of bimanual control where the two virtual arms move independently. So now let me switch gears a little bit, because now as a second variable, I will add a sensory feedback. So in this experiment that we called brain-machine-brain interface, the monkey controlled a virtual hand on the screen, and the monkey task was to scan through a set of virtual targets. So the targets were identical visually, but when the monkey put the virtual hand over the target, it experienced the sensation of a virtual texture, because we stimulated the primary sensory cortex with different patterns of microstimulation, which the monkey felt like vibrations of different kinds. And the monkey's goal was to perform this active tactile exploration under brain control, and to find the vibration that was associated with the reward. And the monkey did perform very well on this task. So let me show you the video. Here the monkey is starting from the center, then three targets show, and the monkey is searching for the correct target. Of course, the monkey didn't have this legend correct target, it is only for us. And the monkey was not listening to the sound either. So the sound is just the artifact of the simulation, which we recorded, but the monkey was not listening to it, of course. The monkey recognized the correct targets through artificial sensations mimicked by microstimulation. You can see that the movements of the virtual hand are a little bit shaky. This is because this is brain control. But still the monkey can manage the task, and it does pretty well. So we published this study, and then we wanted to perform a similar experiment, but with a somewhat more natural way to explore targets, so that the targets didn't feel like vibrating targets, but instead we mimicked the natural tactile exploration of surfaces. So for this purpose, we used virtual gratings. So the monkey had two rectangular squares on the screen and a virtual hand. And then the monkey started to scan these rectangles with a virtual hand, because each time the virtual finger crossed virtual, invisible to the monkey, grating, the monkey received a pulse of microstimulation in its primary somatosensory cortex. So here is how this experiment worked. So the monkey has two gray rectangles. For our convenience, on the right of the screen, you can see green correct target and red incorrect target. But this is only for us, for our convenience. The monkey had just two gray rectangles, and it had to scan it with the virtual finger to figure out the virtual gratings that had higher density. In this particular case, the monkey operates using a joystick, but we could also do the same on the brain control. So in this case, the monkey moves the virtual finger by activating its motor cortex, and at the same time it receives pulses of microstimulation in the primary somatosensory cortex, and the monkey successfully can identify the difference between the virtual gratings. So, and finally, I will describe several types of interfaces that I would call futuristic interfaces, but they also use multiple variables, but this time these multiple variables come from different brains. So we call these interfaces as brain-to-brain interfaces, or brain plus brain interfaces. So here are some examples in monkeys. So here we used 2 or even 3 monkeys, and they cooperatively control the virtual hand on the screen. So in one experiment, we simply added the contributions of 2 brains, and the accuracy of performance improved because errors generated by each brain were cancelled by summation. So the monkey could perform more accurately. In the other task, called partitioned control task, one monkey controlled the X position of the cursor, and the other monkey controlled the Y position of the cursor. Since each monkey had now a simpler task, then they could perform better in the control of this cooperative brain interface. And finally, when we had 3 monkeys, they mimicked something that I would call a super brain, where each monkey performs a task, but it is unaware of the task performed by all brains together. So in this case, each monkey had a 2-dimensional screen and a two-dimensional task for a virtual hand, but all 3 together controlled 3-dimensional movements of the virtual hand. So let me show you how this works. So here, green, red, and blue represent pairs of monkeys, and black represents the contribution of 3 monkeys. Curiously, you can notice that sometimes one of the monkeys could take a break and not perform well, But this design is pretty robust, and then 2 remaining monkeys can take over and perform the task. So besides performing a super task, this interface can also compensate for misbehavior or poor contribution of some of the participants. So, and finally, currently we are running experiments where we have 2 monkeys on wheelchairs in the same room. And 1 monkey is called a passenger because it drives in a motorized wheelchair, whereas the second monkey is called the observer because its wheelchair is stationary. So the driver drives, and sometimes the driver's wheelchair is moved passively. In other cases, the driver can drive itself with brain activity. But the passenger observes the driver, the observer watches the passenger move, and its brain also responds. So we can record representation of wheelchair movement in both monkeys, and then we can build different types of brain-machine interfaces similar to brain plus brain interface that I showed before. So, but perhaps we will have more results on this next year. So I will stop here. Thank you for your attention. And if you have any questions, please ask.
Thank you very much, Mikhail.
You're welcome.
Thank you.
Thank you.
Yeah, I noted down a whole bunch of questions. And before we even get for the questions here, I'm just going to ask you a question that came up on the online forum where people can ask.
Yes.
This one's Wiley, and I think it's really an important question. Did the monkeys have to spend days or weeks learning the 2 robot tasks before the neurons obtained a firing bias? Or did these neurons respond immediately on the first experimental run?
Okay. So it is quite typical for monkey experiments that experimenters spend a lot of time training the monkeys. But actually, it is much harder to train the monkey to control a joystick than to train a monkey to perform in brain-machine interface task. Why? Because when the monkey controls the joystick, then it has to learn that I am supposed to hold the joystick, then move this joystick in different directions. So, which is pretty hard for the monkey. One second. So, but we discovered that monkeys are very good in learning these brain-machine interface tasks because they very rapidly develop this sense of agency for the brain-machine interface task. And in some experiments, the monkeys learned maybe in a week. This is how it worked.
Sorry, I was muted briefly. We have more questions. I'm going to ask one of my questions just for a second before
yes.
others. So this one is, I don't know if this is an easy or difficult question to ask, but I'm trying to get a gist of the overarching insight that we're supposed to get from these discoveries that you've made about how the neurons are encoding things. Or, for example, what is this cooperative super monkey supposed to teach us?
Okay. So this cooperative super monkey is just the first step, I would say. So in the future, I think there may be many applications of different types, from clinical applications where you need a therapist cooperate with the patient to achieve some rehabilitation using a brain-machine interface, to tasks like many people together solving a problem. And of course, some of these problems may not need a brain-machine interface, but some may benefit from this brain interface approach. For example, the tasks when a person cannot really clearly formulate where he is at what stage he is in problem solving. But if you record brain activity, you may get a better idea. But of course, this is all kind of preliminary and just at a speculation level. But I think in the future we will see many interesting examples and many interesting developments. So, like, if you think about mind transfer or mind sharing or mind copying, these may be also very interesting experiments for this kind of tasks, because if you learn how different people could share information, then you may get a better idea of how to transfer mind to, let's say, a computer.
But would it be correct to say that right now the main crux of what you've done by figuring out how to extract this multivariable information, that this is an applied approach? It's about how do you use this for brain-machine interface, say, for example, for a patient who needs to regain some motor control or something like that. It's more that applied approach rather than, say, the analytical approach of understanding, or is it? I mean, of brain coding.
Yeah, I would say it is both. There are many practical applications for this. So basically, we need to extract more information simultaneously, just to get a better control. And practically, this will be very important, because currently the majority of interfaces are just for experimental environments where things are very clearly isolated, but in real life it is very different. So many factors are involved. But also, as we develop all these practical applications, we generate a lot of fundamental results. So it basically goes both ways. So it benefits both clinical science and fundamental science.
Let me quickly ask if there are any other questions here in the audience. I think my audience locally is almost ready for, I don't know, or something. So we're slowing down over here.
I see. Okay.
A long
you had this marathon of talks.
Absolutely.
Yeah.
So I don't see any further questions on the forum either. So in that case, I'd like to thank you very much for your contribution.
You're welcome.
And yeah, I'll give you one more applause for that.
Thank you.
And
thank you.
I will say a few words to close this workshop. Not too many words. Yeah, so first of all, thanks everyone who either showed up in person, prepared to talk, spoke, sponsored us, attended online, and especially anyone who managed to attend all the way from the very first talk through to the very last, because that takes some perseverance. Thanks for anyone who managed to do that, and all of those who did. And then as a final point, just because we are trying to collect some information, I just want to remind people again that we have a survey we're trying to get some people to fill out. If anyone wants to head on over to carboncopies.org/survey, then your answers there would be very much appreciated. And with that, I'm closing the day. Thanks, everyone. This was a great experience, and it's going to take us a while to process everything that we've learned here today. We hope to publish some more about that, and also maybe look at some of the questions that didn't get a turn today and see if we can put some answers for those online as well. Thanks, everyone. Bye.